Take a look at our diagram now, which is going to help us with the differentiation between the male and the female phenotype as we develop. Let's take a look at this beige area first, which is representing the gamete producers, which are also known as the gonads, and our examples here are going to be the testes in male and the ovaries in female. If we direct our attention to the lateral sides of our diagram, I like to call these the tubes. And this is just a simple way of thinking about the male and the female structures that are going to support our gametes throughout reproduction. So you can see in our male, it's going to be things like the seminal vesicles, the epididymis, ejaculatory duct, and ductus deferens, whereas on the female side, we're going to have the oviduct, the uterus, and the upper vagina. And you can think about all these as facilitating gamete transport in the earlier stages of reproduction. Whereas if we move down into our blue area, this is where we are going to have our external genitalia, which will be the structures responsible for intercourse and urination, and also delivery of the fetus when it is time for it to be delivered. So to start, let's talk about the germ layer that's going to be responsible for determination of our phenotypic sex. So if we look here, we're going to start being able to differentiate phenotypic sex around 10 weeks on ultrasound. And remember, it's going to be our intermediate mesoderm, specifically the mesonephros, that is going to help us with determination of these structures. So let's go ahead and take a look at how this happens. So we're going to start out here with our gonadal ridge, which is an undifferentiated gonad. This could become testes or ovaries at this point. And remember, this section of our diagram is all about our gamete producers, so this is going to be our testes or ovaries. So in the case of a male, we are going to have an X and a Y chromosome, whereas females have X and an X chromosome. And this Y chromosome is really important because the Y chromosome contains something called the SRY gene. And the SRY gene is responsible for the expression of a protein called testes determining factor. And testes determining factor does exactly what it sounds like. It produces the testes, so it's going to create testes from the gonadal ridge. So once we have our testes, we have two different cell types within our testicles. Let's start out by talking about our Sertoli cells. So our Sertoli cells are going to be the cells that go on to make sperm later on in puberty. And I like to remember that Sertoli and sperm both start with S. Now, Sertoli cells during development have a really special job. They are going to create a chemical called anti-Müllerian hormone. And what anti-Müllerian hormone does is it's going to go over to our paramesonephric ducts. And remember, on these lateral sides of our diagram, this is all about the supporting tubes, right? So our supporting tubes can either be male or female. So our female supporting tubes are called paramesonephric ducts, also called Müllerian ducts. And what this anti-Müllerian hormone does is it goes up here and it inhibits the support of these paramesonephric ducts. So they actually go on and they regress. And it's important that they regress because if they stay, then you're going to get production of female structures. And we don't want that when we're trying to express phenotypically male. So that's what happens from the Sertoli cells, AMH. We get regression of paramesonephric duct. Now let's talk about our Leydig cells. So our Leydig cells are responsible for producing testosterone, and testosterone is going to kind of have the opposite effect of anti-Müllerian hormone. It's going to have a positive agonism on maintenance of the mesonephric ducts, also known as the Wolffian ducts. So it's the testosterone that's going to maintain these ducts throughout development. And these ducts are going to go on to be the supporting tubes for our sperm. So remember, our lateral portions of the diagram are all about supporting tubes. In the case of our supporting tubes from the mesonephric duct, we're going to get our seminal vesicles, our epididymis, our ejaculatory duct, and our ductus deferens. And I like to remember these with the mnemonic seed. This helps me remember these. So let's go ahead and do a quick review. So remember that our testes are going to come from the primordial germ cells of our gonadal ridge. We are going to have the SRY gene from our Y chromosome. It's going to produce testes determining factor, which is going to lead to the development of testes from our gonadal ridge. It is our Sertoli cells that are going to produce our anti-Müllerian hormone that are going to go up here and inhibit the maintenance of our paramesonephric ducts. And then is our Leydig cells that are going to produce testosterone that go on to keep our mesonephric ducts alive, which creates the structures spelled out by seed. So while it is doing all its work with seed and the mesonephric duct, it's also being converted into DHT, which stands for dihydroxytestosterone. And this is done by the enzyme 5-alpha reductase, and this is an important enzyme on step 1. Just so you guys know, 5-alpha reductase is going to be inhibited by the drug finasteride, which we use for BPH. So that's another plug-in to another part of the reproductive system. But I digress. Continuing on, DHT is going to be what is responsible for the production of our external genitalia. So our external genitalia are going to be formed by the production of DHT, whereas the internal genitalia, as we talked about with seed, are going to be produced by testosterone. So just to review in our bullets here, we have external genitalia development by DHT, and then it's going to be the testosterone that turns into DHT via 5-alpha reductase. So I want to take a moment to do a couple thought experiments to talk about what would happen if we eliminated portions of this cascade. So imagine if we didn't have a Y chromosome. We would not have an SRY gene. And without an SRY gene, you cannot make testes. So pretty much we would halt right there, and we would just default to go over to our ovaries. So that's the case if we were to be without a Y chromosome or without expression of the SRY gene. Alternatively, let's look at a different thought experiment. Imagine we did not have Sertoli cells. If we didn't have Sertoli cells, A, we wouldn't be able to make sperm later on in puberty, and B, we wouldn't be able to produce anti-Müllerian hormone. And so we would have maintenance of our paramesonephric ducts. They would fail to regress. So likely the rest of male development would go on as normal. So all of this stuff would continue to happen. However, we would also have the paramesonephric ducts existing at the same time. So we might have imaging or physical exam findings that showed additional female structures along with the typical male structures. The last little thought experiment I want to talk about is, let's say we had absence of 5-alpha reductase. All development would occur normally from here all throughout here in our supporting tubes and in our gamete producers. However, we would be unable to produce DHT because that needs 5-alpha reductase, and therefore we would probably have failure of production of male external genitalia, and the genitalia would appear female because that is the default, and without DHT, those female genitalia cannot become male. Let's move on to talk about the development of our female phenotype, and it's going to be pretty easy to understand because the male is quite complex, and if you can understand the male, the female is a breeze because it's just the default. So we're back at our gonadal ridge, and let's say we don't have a Y chromosome, we just have our X chromosomes. We are not going to be able to produce testes, determining factor, and so the default is going to be go to ovaries. So we will get ovaries, and then instead of testes, our ovaries are going to be producing 2 other chemicals. So in our theca cells, we're going to get androgens, and these are going to be converted to estrogen. And then in our granulosa cells, they're going to directly produce estrogen. And then our estrogen is what's going to be responsible for creating the female external genitalia. So you'll see that in the absence of testes, it's a pretty straightforward path to the production of estrogen, and then the estrogen is what is going to create our female external genitalia, as well as the absence of dihydroxytestosterone because we are not producing testosterone because we do not have Leydig cells to do so. Additionally, since we never made Sertoli cells because we didn't have testes, we are going to have failure of AMH production, and so we're not going to have inhibition of our paramesonephric ducts. Rather, they are going to maintain themselves, and we're going to get production of some important structures, these structures being the oviduct, the uterus, and the upper vagina. Contrast this with the fact that we have no testosterone to support our Wolffian duct, so that is going to regress. It's not going to continue to be strong. Instead, we're going to have development of our paramesonephric duct. So you can see that differentiation of the female phenotype is largely going to be default from basically whatever didn't happen in the male phenotype. But let's review over our bullet points. So we talked about ovaries coming from the primordial germ cells of the gonadal ridge, and it's our granulosa cells that make estrogen. Our theca cells are going to make androgens, which are eventually going to turn into estrogen, and it's our absence of the SRY gene that allows the paramesonephric ducts to persist, and this was because we never got down to our Sertoli cells with AMH, so we allowed those ducts to persist. Then those ducts were going to go on to make the oviduct, the uterus, and the upper vagina, and it was our estrogen that was responsible for our external genitalia development, as well as the absence of our DHT, which was down here. We do not have DHT because we don't have heavy testosterone production in the female phenotype. Let's take some time now to discuss some homologous structures of our external genitalia. So starting with our genital tubercle, we're going to have the formation of the glans penis and the glans clitoris. So that is going to be roughly this area around here, which I'm going to be highlighting in red. Moving on to the urogenital sinus, this is going to go on to create the prostate gland as well as Bartholin's glands. So that's going to be roughly this area around here. And I would say it's important to note that our prostate is not going to come from seed, and that's an important factor here. It's going to come from the urogenital sinus. This is the one internal male structure that is not coming from our mnemonic seed, and the step 1 exam likes to test on exceptions like that, so I would commit that to memory. Our urogenital fold is going to be highlighted in brown, and it's going to be forming into the ventral shaft of the penis as well as the labia minora. This is going to be the portion that I'm highlighting here roughly in brown. And then finally, we have our labioscrotal swelling, which is going to go on to become the scrotum and the labia majora, which I'm going to be labeling down here in this area in orange. So I want to wrap up by giving you guys a patient vignette, and usually these are going to be presented as some type of ambiguity between either the internal or the external genitalia, and they will oftentimes give you the chromosome count of the patient as well as their sex chromosomes. So let's imagine that we have a patient with XY chromosomes, and on routine imaging for some other health problem, they notice that this patient has both mesonephric duct structures like seed and paramesonephric duct structures like the oviduct, uterus, and upper vagina. How can this be happening, right? This is an XY patient. So we kind of take a look at our diagram here and we'll see that, okay, the paramesonephric ducts were supposed to regress because of production of anti-Müllerian hormone. So it could be possible that this XY person is actually failing to produce anti-Müllerian hormone, and therefore this structure is continuing to persist. However, they're also producing testosterone, so their mesonephric ducts are also continuing to persist. This is an example of how questions like this are asked, and they may ask you at any time point here along our developmental timeline, like where did the error occur or which structures would we expect to be here. So the way to approach these questions is to have mastery over this diagram, and you can ask any question and you can answer any question that you may encounter on test day.